customize and validate especially your protocol in terms of the wet lab work. So we have to validate what is the percentage of recalling of the variant among the population, either using low coverage or high coverage. And we find out if we have approached something, we call it intermediate coverage. I mean, instead of going for high coverage, for people who are familiar with next-generation sequencing protocol, 30x coverage is a little bit high coverage. With if you are going to low coverage, you need 5, 6 coverage, something like this. But we are optimizing and optimizing our protocol to at least to have between 15 to 20 coverage for our gene. The whole exome is to start in parallel as well for the same individuals and same subject that are involved in this project, and matching both data from the whole genome and whole exomes together and validating the findings, especially in the coding region for our subjects that they are involved in this project. In terms, we have very good actually quality data in some at the beginning. Like if you can see at the far end of the slide, I'm not sure whether this is working or not, but it's more than 90% here. One of the subjects, it's a little bit low coverage, but still at least we can recall some of the variant for this one. If you go for the whole exomes, you can see all of our subjects are with low coverage going. The recalling rate is more than 91%, which is give us, let's say, good hoping or good warming into keep going for this project. This slide actually summarizing some of the statistical outcome of the project, and you can see from this one, this project, it's like a preliminary data from this project, and you will see later on how this variant, when you add more subjects into this project, you see this, we have almost 280,000 novel SNPs compared to the reference sequence. Actually, we combine both exomics and genomics sequencing using next-generation sequencing. Some of this has been also validated using some of the third-generation sequencing like ion read technology and gender sequencing as well from Roche. If you can see, this is an example for one of subjects that has been sequenced long time ago. This project, by the way, has been started 2008, and due to logistic and infrastructure issue and other issues, it's been on hold almost 4 years. We start again this project 2012, and you can see this subject has almost more than 450,000 SNPs that different and novel compared to other sequencing like venture genome. This is a little bit old genomics database or reference sequence. And when we compare it also to the SNPs database, it shows that this subject sharing a little bit in the database. I mean, you can see almost 1 million SNPs found already and existed in the SNP database, and a little bit with venture genome, almost 33,000 SNPs that have been found in this subject that shared with the venture genome sequence. When we compare our two individuals in this subject, you can see they are even among the individuals, they have high discrepancy between them. And you can see they are sharing here almost 600,000 SNPs, while they are different in 51,000 SNPs. So this means, or this is highly strong very strong evidence that each individual has different and unique genome that will help if we know every single individual, especially for patients, how these patients can be classified into a group in terms of treating the patients. And I guess that this will be a very strong infrastructure for personalized medicine practice in the future. And you know, this is going on worldwide and start actually our running here in the kingdom. This is as example of one of the SNPs. If you can see the reference sequence here in the bottom, and you can see it's a G nucleotide, and if you compare the subject, if you compare it to the reference sequence, you'll see that this subject has a SNP, which is different. If we sequencing, this has been actually analyzed, and we found out this SNP is existing in non-coding region, which is not affecting any protein activity in this region. This SNP has been found in chromosome number 1, while we analyzed a deletion event happened also in chromosome 1, and you can see when we recall the contigs for this sequence, it's hard to find the breakpoints of the small fragment which is in the middle. I'm not sure that the pointer is okay, but maybe yes, I can use this one. It's this one. They're both breakpoints. It's highly to be recalled when we analyze this individual. So this means that this fragment is deleted from both copies in this subject. Again, this deletion is happened in non-coding region for this subject. An inversion event happened in chromosome 4 in the same individual. This one again is affecting a single gene, and this is a little bit high or large inversion, which is affecting around 8,500 base pair. And again, we find we cannot recall the contigs in this region, so that confirm that there is some deletion. We validate this using some of the microarray, CGH microarray, and confirm that this part or this fragment is also deleted in the individual. Okay, our experience in how many persons or how many persons that we can enroll them in this study. Actually, it's very difficult to recall the genome of any population only from one individual or 2 individual or 3 individual. I think we need lots and lots, even though each individual in the community has his or her own genome features that a little bit different from the other. If you see this, another subject actually, which is the first subject that has been analyzed, and when we compare his genome to the phase 1 and phase 2, the HapMap database, we found out that it has 1.4 billion SNPs, while when we match it or align it to the phase 3, it's dropped more than half. It's 670,000 SNPs. When we compare it to if you see it in the venture sequencing, you see the Arab sequence is a little bit similar in terms of number of dropping when you increase or when you go and align alignment into the phase 3 for the HapMap database. This individual actually has a high rate of sharing with other population. Actually, there's no big difference difference between Caucasian or Japanese, Chinese, you can see from this figure here, and in European tribes, which is African tribes here. While when we combine the data from different, from more than one subject, you'll see this picture is a little bit different. This is just giving you an overview when we see how the shared distance between Saudi individual and other populations. And you can see there is some similarities in terms of number. Most of them is around more than 1.2 million SNPs that we shared with other populations. How we analyze this one, this is a bioinformatics slide actually, but it's simply, I try to make it as simple as possible, that all the data being generated from next generation sequencers, and the main platform that we are using is Solid 5 from Live Technology. So it's been sent to our clusters and try to map them and pair them into together. Then send them for a variant calling format. We are using now at the moment a couple of bioinformatics softwares, which is Celsi Bio and LiveScope platform. In addition, now we are working these days on adding genomics as a third tool for our bioinformatics analysis, to have more validation and more precise answers into our finding. This is when we combine the results from more than 32 subjects, because now we are completing 32 subjects, and our target at the moment is 50 subjects, and I think that by end of this year we are going to complete these 50 subjects. But if you can see this, the most important part in this slide, this is busy slide, that we are unique in 1.9 million SNPs, that different from other populations, while the closest tribe to us is the European tribe, the African tribe here. We have lowest sharing with the Caucasian population. This is again from 32 subjects, and you see that this picture is completely different from what you saw a few slides back from one subject. So our experience suggests to add more and more subjects into this project. Possibly, maybe if we go into intermediate coverage for this project, we may approach a little bit good picture into our genome when we complete 250 subjects. Just a small comparison indicating the variation among chromosome as a chromosome in whole, and we compare it between NCBI, reference sequence, and Celera sequence. This is just showing you only selected chromosome 3, 12, 19, X and Y, and you can see there is any variation or differences between both reference sequence. It's not that significant, except if you compare X to Y chromosome. Surprisingly, it's not surprisingly actually, but it's since chromosome number 1 is the largest, we expect that we will find the SNPs counts or SNPs number are higher in chromosome 1, but we found it out in chromosome number 2 here. While in the heterozygosity or heterozygous SNPs in X and Y, we couldn't find any heterozygous SNPs among these two chromosomes. This is again in one individual, and it's existing in most than one actually individual. While the picture is a little bit different when we go to the deletion insertion polymorphisms, you can see here that the homozygous here in X and Y is not existing, which is actually surprising us, and this needs also more work to validate and see the effect of this homozygosity, especially on chromosome X. This is just again overview for all chromosomes and indicating the large polymorphisms, deletion, inversion polymorphisms, and you can see that when we compare it to the reference sequence or the SNPs database, you see most of our frequencies are very high, more than 97% actually, in terms of the accuracy, and all this data has been validated again using other methodology and other technology. This is the SNP distribution across the genome, and you can see from this slide that chromosome number 2 is the highest one, has in this type of variant, while chromosome 1 has the highest non-synonymous change, which is also need more actually study and try to find out this non-synonymous alteration, where it's happened and what's the cause of this one. So the functional assays for studying all these non-synonymous is a huge task, and it needs actually a national efforts, not only from one institute. It needs multi-institutional work. In terms of protein interaction and strings association in this slide, we found out that our population has some unique protein interactions in some of the genes, and we put some example here, X1 and RBL13, MAP2K3. This is unique in our population compared to the other gene that we share with them the same feature of protein-to-protein interaction. This is actually just an example of one of the genes that has more than 3 protein-to-protein interaction activity among its network, and it's been analyzed as a soft copy also to study in this using string database for protein. You have 2 minutes to go. I'm finishing right now. Okay, this is again summarizing what is the structural rearrangement, including large deletion, insertion, inversion, all these things from our 32 subjects actually. We accumulate, or this is inaccurate actually, and this is the validation that we've done through for some of our finding using Affymetrix platforms as a tool of validation. Finally, actually, the mitochondrial genome, we have find our genome is missing 1 base as a deletion. Why? There is a 46 different SNPs or 45 SNPs that are different compared to the reference sequence, NCBI reference sequence, and our one subject has been uploaded and published in the NCBI gene bank, and there is now 48 mitochondrial genome are in submission process to the gene bank, and we hopefully by the next few months you will have these published and available to the researcher. I would like finally to thank Dr. Al-Askar, our Executive Director, and congratulate him for his new appointment as Executive Director of the KMARC. My appreciation for Dr. Mohammed Al-Jimna, our former Executive Director, for his unlimited support for this project. Also, I would like to thank Dr. Mohammed Al-Louis for his day-to-day supervision of this project and close training for our staff. Actually, this is our staff, actually, and this is our pleasure, actually, that they are well trained in this technology. I like that the operational lab, they make our life easy. The biobank team, they produce actually excellent materials for the exterior and facilities database. Actually, we are all proud of them in the KMARC, and my special thanks to the team that they make the effort to here at the present our booth, which is Mr. Zaki Rabhan, Bnayan Al-Jani, Mrs. Nawf Fateri, Balqis Al-Hamad, and Warda Al-Habbi. Thank you very much for your listening, and again, thank you for the organization committee for inviting me.
I think for the sake of time, because we are running behind in the whole, we will defer all the questions during the break. So we will break for coffee and the prayer now, and then we will resume with the next session to come. Thank you.
Okay, welcome to the next session. And so we have some great speakers for this session. The first is Professor Apollo Sassoni Corsi, who will be speaking on the epigenetics and metabolism, the clock connection. So Apollo is a professor and chair of pharmacology at UC Irvine in California. Prior to that, he was director of research at Strasbourg in France. And he's really made several contributions to our understanding of cellular signal transduction and mechanisms of control of gene regulation, including chromatin and histone regulation. And he's really a leader in the genetic control of circadian rhythm, including key regulatory events in histone regulation. So we'll welcome him, and you have the podium.
Thank you all. It's a pleasure to be here. I'm going to tell you a few things about the studies we've been conducting in our Center for Epigenetics and Metabolism, occupies the top floor of this building at UCI. This is my group, and what I'm going to tell you today mostly is work done by, I guess, two ladies. This one right here, Christina Kelmahan, and this one right here, Summer Masri. And we've been lucky enough to have the support by this fellow right here, sabbatical professor from Munich. His name is Axel Niemoff. All right, so the key question that we'd like to address in our center is how nutrition impacts on epigenetics. And I'll tell you very briefly about what is epigenetics in the introduction of my talk. The question about nutrition is critical, especially this time in most countries where epidemics of obesity and diabetes becomes dramatic. This is true specifically here in Saudi Arabia, as you can see from these slides. Saudi Arabia is one of the countries where obesity is at the maximum and is increasing with dramatic proportions. In fact, the rate of obesity increase in Saudi Arabia is higher than any other country in the world at this point. And this is the rate of diabetes. As you can see also, Saudi Arabia is in a bad situation. So I think the questions that we address in our center are highly relevant to this country. One really sad record of Saudi Arabia is the report of the very first, oops, okay, I didn't do anything. Maybe I shouldn't talk about that stuff. That's why it isn't here. Okay. Is the record of having a very first 2 years old obese child who went under a gastric bypass surgery to decrease the rate of obesity and diabetes. Now, what is that determines all this and how this relates to epigenetics and specifically to the disruption of the circadian clock? That's what I'm going to tell you today. Now, the concept about epigenetics comes from this fellow. This is was an English developmental biologist, Carter Waddington. In the 50s, he came up with, okay, I don't know what to do, but I'm not going to touch anything. All right. He came up with the concept that equal cells with the same genome will become different And this is the basis of all stem cell biology these days. How is it possible that two equal cells will become either a neuron or adipocyte? The idea is that the landscape will be different valleys and troughs in a specific landscape where the cell, here represented by marbles, will roll down, and the landscape will shape their genome so that they will function in different ways, and thereby determining the fate of those cells. How is that possible, and why is so important that genome? I'd like to show this slide. This fellow is Kobe Bryant, and he trains at UCI, so I kind of meet him all the time in the coffee shop. And one day I realized that he's as tall precisely as much as DNA we have in one cell. So in one cell we have 6.5 feet of DNA, 2 meters, and that's how much Kobe is tall. That means that in his retina, he requires to shoot the ball in the basketball is the same DNA that he has in the muscle that gives the power. But how is it possible that DNA will turn out to be so different as a fibroblast or a neuronal retinal cell? This is based on one thing, which is the genome that is way more complicated than just 2 meters of DNA. DNA is organized in chromatin, and those 2 meters contain large amounts of proteins. And these are organized in the nucleosomes, shown here. Nucleosome is based on 8 histones, 4 dimers, and please note these tails down here sticking out from each single histone. I'll talk about them in a minute. DNA wraps around every single nucleosome twice, and this happens every 147 base pairs all along the genome. The question is, while this compaction of DNA occurs and then superstructure is organized, highly dense, the question is, how is it possible that we have physiologically meaningful and time-specific activation or silencing of gene expression? So a large amount of epigenetic control goes through chromatin. This organization is called chromatin. And we wonder about what are the mechanisms that allow chromatin to be either open or closed for gene expression. So you remember those tails I showed you. This is an example of a tail of the H3 histone, one of the histones composing the nucleosome. And this is the tail sticking out from each single group of proteins. Now, these are platforms where a number of post-translational modifications can occur, either acetylation, methylation, phosphorylation, and so on. And the presence of those specific covalent modification plus their reciprocal organization will define whether a specific site will be either open or closed for transcription. And I'm going to go through these details. Is not the point here, but what I want to tell you as a concept is the following. What are the enzymes? There are enzymes doing the job of adding an acetyl or a methyl group. These are called either writers or erasers. That's the way we like to call them. A writer will add, let's say, a phosphate group or acetyl group or methyl group as examples, and thereby these enzymes will be called acetyltransferases, methyltransferases, or kinases. The eraser will do the reverse job of removing those, and thereby called HDACs, demethylases, and so on. There's another group of proteins called readers. These generally don't have enzymatic activity, but what they do, they recognize those modifications through specific domains, Brown domains, chromodomains, and so on, and through those will connect to the rest of the nuclear machinery, let's say including the transcriptional machinery and so on. So the concept number one I want to convey to you is the following. These tails are like platforms where all these modifications can occur, and through those modifications, we change a specific locus of the epigenome to be open or closed for gene expression. And then these are the enzymes, but those enzymes, in order to do that job, to add the acetyl groups, to add the methyl group, they need metabolites. So the concept I like to convey to you is that the epigenome actually senses cellular metabolism and is able to use for its own purpose, by modifying tails, the amount of metabolites in the cell. Let's say you need a lot of acetylation. You will use a lot of acetyl-CoA around, it is a major source of acetyl groups in order to obtain those acetylations. Well, where those metabolites come from? They come from these guys, metabolic pathways, which I'm sure you all know well, the Krebs cycle, the urea cycle, glycolysis, so on. So what we did in this slide is to add a star on every single enzyme that is known to be working in a circadian manner. And as you can see here, basically every single metabolic pathway is circadian. Now, what is circadian? The word circadian comes from Latin. It means a circadian. Those are cycles of about 24 hours. And they're highly conserved all the way from cyanobacteria, plants, invertebrates, and of course vertebrates. So they're extremely conserved through evolution. And they use Zeitgebers, this is a German word, means time givers, in order to be in trainer readjustment. And the most important Zeitgeber is light. That's exactly what's happening to me, let's say, since I have 11 hours of jet lag in California. And my internal clock is adjusting to the Saudi Arabian light-dark cycle, which is kind of a difficult thing to do, but I'm trying my best. Now, from a molecular standpoint, the clock is organized in general by a transcriptional translation feedback loops. Then I'll tell you in a minute. What is really interesting is that those loops are highly conserved structurally and sometimes only conceptually between species. And the genes that are involved in the clock control have been cloned. The very first one was per period in the fly by Seymour Benzer in the 70s. And then after that, many other genes were cloned in mammals and other species. And the key thing is that what they control a large number of clock control genes of CCGs. Where is the clock in mammals? The central clock is in the brain, and light hits the back of the retina, eating specific cells called ganglion cells. And the light signal travels the retinal hypothalamic tract all the way down to the hypothalamus and controls this group of neurons called the suprachiasmatic nucleus. There are about 20,000 neurons like this in every mammal. These neurons have the amazing capacity of being oscillating in the 24-hour cycle. No matter what, you can place them in culture, they will still oscillate. Now, additional experiments by a number of laboratories have shown that in reality we have clocks everywhere, even in the periphery, in liver and heart and so on. And what we know today is that there's like an orchestra of clocks in every body, in every organism, where the SCN in mammals is the central organizer and synchronizer for the peripheral clocks, which, however, can be uncoupled from the central clock by various ways. One is alternative rhythms and activity, or either restricted feeding, like let's say what I'm doing right now by having lunch at the wrong time of day for me. All right, so what is the basic of the clock and how it works? So this is now from a pure molecular standpoint. You have transcriptional activators called clock and beam out, and they direct the expression of repressors called purr and cry. Now they will fuse now together just for simplicity. You'll see that happening in a second. There you go. So clock and beam out will transcribe purr and cry. And this is now daytime. This is the sun. Purr and cry will encode the repressors, and these repressors will dimerize and then migrate into the nucleus. So you have a number of possible levels of regulation. One is transcription, of course. The other one is translation, and there nuclear translocation. By the time all the repressors are in the nucleus, it's nighttime, it's the moon. So what happens is the repressors will shut off their own expression by blocking the activators. Then there's another level of control. You see that there's a specific disruption and destruction of the repressors, and this happens through a specific program of a proteasome. They're F-box proteins specific for the clock system. By the time all the repressors are gone, it's daytime again, and this happens every 24 hours in every cell in our body. So what is happening when you destroy the system, when you destroy this beautiful molecular machinery? Lots of problems. You have increased insomnia, depression, metabolic disorders, inflammation, obesity and diabetes, even examples like accelerated aging, and of course even cancer. So you can see that disruption of the clock is a problem. Now, yes, there's a lot of information about when you disrupt the clock system by mutation in the mouse.
you can see all the different type of metabolic disorders you have. So how this works and why this is important? It's important because we believe that the connection between the clock and metabolism is not only transcriptional, but also is epigenetic. And this was shown by this particular group of experiments I'll show you very quickly. So the question is, we have something like 15% of our genome that is controlled by the clock system. And this goes through, in generally thought, to go through E-boxes. This is the most common promoter element in the genome. So what really happens is the oscillation of gene expression, and thereby beautiful harmonic cyclic expression through a mechanism of gene expression. However, as I told you, 15% of the genome is a lot, and we thought had to be connected somehow to changes in chromatin level. And this is an experiment that started several years ago by a postdoc, great postdoc from Japan, Masao Doi. Masao, what he did was to take now an antibody, well, recognize the tail of H3 only when it's acetylated in K9 and K14. When he uses that antibody and recognize the specific site, you will see that on a promoter of a gene that is normally oscillating like this one, and that acetylation of that tail also is cyclic. And what he discovered that the enzyme doing that job actually is clock itself. So clock, what we thought being a simple transcription factor, is also writer, is also histone acetyltransferase responsible for the acetylation at circadian sites. Additional experiments by a number of other fellows, Junior Oyama and Yasu Nakahata, I have no time to go too many details, but found that in reality the clock system, all right, is connected to another really interesting controller of chromatin, and this is an HDAC, so an eraser called SIRT1. Now, SIRT2s are really interesting HDACs because they respond to changing level of NAD in the cell. Well, we thought at the time that we have a complex that is clock and SIRT1 together, whether the balance will go towards the writer, we have acetylation, and thereby active transcription. Whether we go towards the SIRT1 level, we have silencing because the deacetylation event. Additional experiments by Yasu found also that in reality, when you look at the acetylation changes, believing that only clock and SIRT1 are implicated was a problem, because when you go and look at the levels of clock and SIRT1, they generally do not oscillate in most cells. So how is it possible we have oscillation in the readout of acetylation, while instead we have basically no changing in the protein level? And that comes to the problem of metabolomics and metabolites. Well, we found that it's really NAD that is oscillating. This is the metabolite NAD that is oscillating that then tells the protein SIRT1 to start to be oscillating in terms of enzymatic activity. And this was what we did, and thereby we connected the enzymatic feedback loop of NAD synthesis to the transcription feedback loop of the clock system. Now, at the same time, more or less, another postdoc in the lab, Kristen Eckelmahan, asked the question, if NAD as an enzyme, as a metabolite is oscillating, how much of the metabolome is oscillating? And this connects to the talk by Dr. Fornassi this morning. So what Kristen did was to take now liver samples at different time of day and night and did a metabolome by mass spec. And we were able to read something like about 600 molecules, 600 metabolites. And you can see different groups of metabolites will have different types of peak during day and night. Whether you look at amino acids, nucleotides, carbohydrates, lipids, or xenobiotic metabolites, you'll see different type of peaks. All this was a huge amount of information that we were successful in putting together into some type of form. But the most important thing to say is that what we found is that of all those metabolites, about 50% of them oscillate, which is really remarkable. I mean, it's like we have half of our metabolome that is not flat, but is oscillating in a cyclic manner along the circadian cycle. And using the specific knockouts for the clock system, we could also see that a good chunk of this 50% of metabolite oscillation is clock-driven. Now, I don't have to go through all the situation here, but all this is now in a website and a resource that is called Genomic. You can go and take each single metabolite of your interest in a number of tissues and know whether it's oscillating or not oscillating. whether that oscillation is clock driven or not. So I strongly invite you to go and do this. And you can do like NAD, uracil, whatever you like. And what we found is that we could look at the specific oscillation of metabolite and connect a specific metabolite nodes to specific transcription nodes and find that there's a coherence between those two. I don't have the time to go through all those details, but I can definitely tell you more about later on if we want to discuss. I take these last few minutes to tell you another story which is going to come out very soon, and I thought could be of interest specifically to this group of people. It has to do with nutrition and has to do with the problems of diabetes and obesity. And this has to do with the concept of reprogramming of the circadian clock by nutritional challenge. The paper is going to come out, I think, in 1 or 2 weeks in South. Now, the thing is the following. How much when we eat different amount of food or different types of food or different time, this will change our clock, and is the clock able to adapt through its own intrinsic plasticity to it? Well, we've done one experiment that is designed here. What we did was to take mice in normal, chow, or high-fat diet, and this is done for about 10 weeks. And then we take the mice at the end of this 10 weeks of either normal, chow, high-fat diet, and we sacrifice them every 4 hours along the circadian cycle, covering a cycle and a half about. And then we took all samples you want. We took liver, muscle, serum, and so on. And then from those, we done the transcriptome and metabolome analysis by mass spec. Of course, I have no time to go and tell you all the data we got, but just to give you a couple hints about the results we obtained that I believe provide some really interesting insight. Well, first thing we can show you is that when you go and look now at groups of metabolites that oscillate in normal chow versus how they oscillate in a high-fat diet, you can see just visually that when you take groups of metabolites of mice being under high-fat diet, that dramatically switches the groups of metabolites that will oscillate at different time of day and night. This now doesn't tell you a single metabolite when is oscillating. This tells you how the abundance of metabolites at different time of day and night. But of course, we can tell you precisely each metabolite what it does. And again, I don't have the time to go through all the details, but you can go and look at a couple of examples as shown here that I think are really revealing. Well, I don't have to tell you all this. This is now normal chow, high-fat diet, different time. There's diet gavers along the circadian cycle, and you can see all different nice metabolites which will oscillate. I think the top here is sorbitol, and then stops to be oscillating in high-fat diet. But a couple of examples I think are really revealing here. One is NAD. I told you NAD is controlling sirtuins. So you're controlling HDAX, is controlling the deacetylation of part of the genome. And yet, when you put this mice in high-fat diet, you can see that while in normal chow, you have the normal nice oscillation of NAD that we already reported. That oscillation is completely abolished when you put mice in a high-fat diet. So what you do when you have a high-fat diet, let's say you have too many burgers, too much pizza, that goes for too many days, what you're really doing, you're disrupting not only gene expression, but you're disrupting also the control of a number of enzymes like the sirtuins and NAD-depending enzymes.